students to my new fresh episode on topic biology. So I have already uploaded some videos on biology also. So if you missed my previous episodes, you can subscribe my channel Physics Scholar and you can see those videos. So in my previous videos I have discussed about what is RBC, what is actually hemoglobin, the function of that, and the diseases, okay, what due to the lack of hemoglobins, what are there, what are the different type of anemias, and also the WBCs and different component of the WBC cells. So the next component, very important component of blood is, that is platelets or thrombocytes, okay, thrombocytes or platelets, that is also very important component of blood, okay. So now what is platelets? So we have learned about the function of RBC, function of WBC. Now today we will learn about the function and what is platelets, okay. So platelets is nothing but it is a blood component which helps for blood coagulation and don't have any nucleus in it. Clear? So what is platelets? So platelets is a component, very important component of cells of blood, which actually helps blood coagulation. That means if any injury is there inside or outside of our body, then the blood after some time the blood will be coagulated. That means the bleeding will stop and we will see the coagulation of blood. So this actually the platelets help for blood coagulation. That is the main function, okay, of platelets. How the blood coagulated and what is the thermoplastin will secrete, that I will discuss later, okay. So that is known as the platelets. So it is a blood component which helps for blood coagulation and don't have any nucleus in it. So next one is the lifespan of platelets. Lifespan. Basically platelets, the lifespan is 5 to 9 days. So within 5 to 9 days the platelets cells will die, okay. So that is the lifespan of the platelets. Clear? So now next, move to the next topic. From where the platelets actually generated, okay, source. So in our bone marrow there is a cell which is known as megakaryocyte. Megakaryocyte. What is the name of the cell? Megakaryocyte. Megakaryocyte cell is there. So through this cell, just like the amoeba, they have pseudopodia. Okay. So the megakaryocyte cell, they have the pseudopodia. So through pseudopodia the platelets actually develops or generates, okay. So in our bone marrow we have a cell which is known as megakaryocyte. So from this cell they have pseudopodia. So from there the platelets actually develops. So platelets is a important cells of our blood, clear, and it helps for blood coagulation and it also don't have any nucleus in it. The average lifespan of platelets is 5 to 9 days, and the source of platelets in our bone marrow, there are megakaryocyte cell, and in megakaryocyte cell there are pseudopodia, and from that pseudopodia the platelets are developed, okay. So now let us move to the next topic, function of platelets, okay. So let us move function of platelets. So what is the function of platelets? So one by one we will discuss. Clear? What is the function of platelets? Function. function of platelets. So first I told already, blood coagulation. Blood coagulation. So the blood coagulation process I will discuss later on in this episode only. Okay, so first I will only discuss the basic thing. So when there is any injury, inside or outside our body, then what will happen? The platelets will break. That platelet cells will come through blood, because the blood is flowing in our blood vessel. So it will break. Clear? It will break, and thromboplastin will secrete. Okay, will secrete from the platelets. The platelets will break down and thromboplastin will secrete. So the thromboplastin actually will first create thrombin. Okay, and prothrombin is there inside our blood, and it will react with prothrombin and create the thrombin. And thrombin, fibrinogen is there in our blood plasma. Fibrinogen. So thrombin will react with the fibrinogen. So thrombin will react with what? Fibrinogen. And it will fibrin. Okay, so it will create fibrin. Clear? So fibrin is nothing but a net-like structure. And the other blood cells or corpuscles, they will what? The net-like structure will be there, and the blood cannot come out from it. Okay, and it will convert into a semi-liquid state, jelly-like state. So that is known as the blood coagulation. Okay, so I will discuss the process of blood coagulation in details. Okay, so this is the first function of platelets. So what is the first function of platelets? First, blood coagulation. So if any injury is there inside or outside the blood vessels, okay, then what will happen? Then the blood will come and the platelets will be broken down or will break down, and what will happen? Thromboplastin will be secreted, and that thromboplastin helps the blood to be coagulated. There are some stages how the thromboplastin prepare the fibrin. Okay, that we will discuss later on this episode. So second function is repair of capillary endothelium. Repair of capillary endothelium. What is capillary endothelium? So our capillaries. What is capillary? What is blood vessels? Okay, later on we will discuss in details. So blood vessels means, I am discussing, blood vessels means the tube through which the blood can flow. So this is known as the blood vessels. Blood vessel basically artery and veins are there. Two type of blood vessels are there, and they also have the capillaries. Capillaries means the branching. Okay, very small pipe like structure. Okay, the capillaries. Okay, the branching just like a net. So what happened? Platelets helps of capillary endothelium. Okay, to repair. That means what? If there is a rupture, so these are the capillaries. Capillaries are like this. Okay, if there is any rupture, that means the wall of the capillaries there is any rupture. That means what? The blood will start to come out, because inside capillary what is there? Blood. So if any rupture is there, then what will happen? The blood will start to come out. Then what is required? Immediate what? Immediate repairment of this is required. So the platelets actually help. Platelets helps to repair the capillary endothelium. Okay, that means if any rupture is there in our blood capillaries, then the blood will start coming out from it. If it is not stopped, then what will happen? So the bleeding will start and all the blood will come out from that rupture. Clear? So that's why the rupture should be repaired immediately. So the platelets actually help to repair this rupture. Okay, so the first function of platelets is blood coagulation. if any injury is there inside or outside our body, then the blood will be coagulated so that all the blood should not come out from our body. Clear? The second one is repair of capillary endothelium. Clear? Endothelium is the layer, okay, a very soft layer inside our capillaries where the blood is flowing. So if due to any rupture, what happened, the high pressure or any rupture, if the blood started coming, then the platelets helps to repair these capillaries very fast so that the blood cannot come out from it. Okay, so that this is also a very important function of platelets. So coming to the next. So next point will be another important function of platelets. Clot serotonin. Okay, clot destruction. What does it mean? So, platelets will secrete serotonin. Okay. So, what is the function of serotonin basically? Okay, I will discuss in details also. Serotonin, just in the function of platelets, I am giving you an overview. So, suppose this is the blood vessels, and due to any rupture, these are the clotting of bloods. So, what will happen? These are the clotting of bloods. So, what will happen? The serotonin, platelets also secretes the serotonin. So, what it will do, the serotonin, this part will be squeezed, that means the volume will be decreased. So, it will be just like this, okay, it will be squeezed. So, this portion will be, the volume will decrease. So, what will happen? It will be contract, then what will happen? And it will flow, okay, flow from the blood vessels, and when it will be contract, okay, or squeezed, then what will happen? Yellowish color fluid, yellowish colored fluid will come out from it, which is also known as serum. What is serum actually? That will also I will discuss. Okay. So, this is what the serotonin. So, what is the function of third function of the platelets? So, platelets will secrete serotonin, a very important thing, serotonin. So, what serotonin will do? If there is any clotting of blood, okay, accumulation of blood is there. So, what will happen? It will squeeze or it will decrease the volume of it, so that it can easily flow through the blood vessels. Clear? And blood can also flow through it. Clear? So, what will happen? And when it will be squeezed or it will be volume will be decreased, then a white color of yellowish color of fluid will come out from it, okay, which is known as serum. Clear? So, that is the third function of platelets. So, hope you have understood. So, three functions, okay. First one is it will help to coagulate the blood. Clear? Second one is what I already discussed, the second function of the platelets to repair the rupture of our capillaries, blood capillaries, and third one is it will secrete the serotonin, which will actually squeeze or decrease the volume of the coagulated blood, so that the other blood can flow through it and it can be also transmitted to the blood vessels, okay, and through that the yellowish part fluid is can come out or will come out, which is known as also serum. So, students, hope the function of platelets you have understood. So, next, let us move to the next topic. Okay, students. So, now if the numbers of platelets increases than normal. So this is known as thrombocytosis. Thrombocytosis. Clear? And if the numbers of platelets decreases than normal. Basically, during some disease, dengue, okay, this happened. The platelets fall down. So it is known as purpura. Purpura. Okay, this is known as what? Purpura. So, two things may happen. First of all, the number of platelets can increase. So it is known as thrombocytosis. Okay, and if the number of platelets decreases than normal value, then it is known as purpura. Clear? So remember these two terms, thrombocytosis and purpura. Clear to all of you? I hope this very common term, what is thrombocytosis and purpura. So increase of number of platelets than the usual is known as thrombocytosis, and decreasing number of platelets than normal is known as purpura. Clear? So next, move to the next topic. Clear? So students, let us now move to the next topic. Okay. So now the platelets is there inside our blood vessels or in our blood, and the blood is flow and it is responsible for blood coagulation, but still the blood cannot be coagulated inside our vessels. What is the reason? Okay, so why the blood can't be coagulated inside blood vessel inside blood vessels. Okay, so first point is, in plasma we have heparin. Now heparin is a anticoagulant. That means some substances are there which will prevent blood to be coagulated. What are the substances? Heparin, hirudin, citrate, oxalate salt. Okay, so these are known as the coagulant of blood. So heparin is there. Heparin is present in plasma which is anticoagulant and prevent blood coagulation inside blood vessel. Okay, so that is the first point. Heparin is present in plasma which is anticoagulant and prevent blood coagulation inside blood vessels. Clear? So number 2, what? Inside our blood vessels there is a continuous flow of blood because our heart expanding and contracting continuously. Okay, I will discuss the function of heart and all the components of heart and their functions in details. Okay, when I will start the heart chapter. Clear, or the topic. So what will happen? Inside blood vessels there is a continuous flow of blood. Clear? Inside blood vessels there is a continuous flow of blood. That's why it will not be coagulated very easily. Okay. Now number C is what? The wall of blood vessel is very smooth. That's why the platelets can't be broken. Okay, so that means what? The wall of the blood vessel is very smooth. If there is any rupture, then the platelets will break and will secrete the thermoplastin. Clear? And the coagulation process will start. Okay, how the coagulation will be done, that I will discuss. Okay, so this is the major 3 points why the blood will not be coagulated inside our blood vessels. The first one is what? Heparin is present in plasma, which is anticoagulant and prevent the blood coagulation inside the blood vessels, and also there is a continuous flow of blood inside the blood vessels that will prevent the blood to be coagulated. And third one is what? Third one is the wall of the blood vessel is very smooth, so if any rupture is not there or any injury is not there, then the platelets cannot be broken and the thermoplastin will not be secreted and the blood coagulation will not take place. Now one question has come or may come in the exam, okay, very often. First, let us discuss what is blood coagulation. Then I will discuss these questions. Okay. So first, let us discuss what is blood coagulation. What is blood coagulation? What is blood coagulation? Clear? So, if there is any injury inside or outside, inside or outside of our body, then the blood will stop flowing, stop to come out from that injury after some times, and it will, and there will be a semi-solid jelly-like structure or substances, jelly-like substance will be created. This is known as blood coagulation. Okay. So if any injury is there, that means if it is not a very big injury, then automatically after some time what will happen? The blood will stop come out, and you see that there is a semi-solid, okay, jelly-like substance will be created, and the blood will not come out from that particular part. So this is known as what? So this is known as, suppose it is the blood vessels, and suppose there is the injury, okay. So what will happen? So after some time, this jelly-like structure will be formed, and the blood will stop to come out from it. Okay. So this is known as blood coagulation. Clear? This is known as blood coagulation. Clear to all of you? Okay. Now, what is the time? What is the time? If we put the blood inside a glass tube, then the time for blood coagulation is 3 to 17 minutes. But actually, blood will be coagulated in 3 to 7 minutes after injury. Okay. So blood will be coagulated in 3 to 7 minutes after injury. If you keep the blood in a glass tube and keep it like that for some time, then within 3 to 17 minutes it will be coagulated. But actually we can see after a small injury, it is not a huge deep. So the blood will be coagulated within 3 to 7 minutes. So that is the coagulation time required for the blood. So now one that I already told, that one very common questions comes in the examination, that if the leech bite us, okay, you have seen the leech, I hope you all have seen the leech. If the leech bite us, then what will happen? Then the blood will not be coagulated very easily. Okay, why? If the leech bite us, then blood will not be coagulated, be coagulated very easily. What is the reason? What is the reason behind it? So to discuss these questions, first we have to understand what is anticoagulant. What is anticoagulant? What is anticoagulant? So anticoagulant are the substances which prevents or stops the blood to be coagulated. Okay, heparin, that is already there by nature, it is there inside our body, inside our blood. Okay, so that is what anticoagulant. So what? The substances which prevent blood to be coagulated is known as anticoagulant. What are the examples of anticoagulant? Examples of anticoagulant: heparin, hirudin, citrate, or oxalate, salt, etc. So these are the examples of anticoagulant. The substances which prevent the blood to be coagulated is known as anticoagulant. Okay. So in case of the leech, if leech bites us, we actually don't realize, and when the leech complete, then it will fall down from our body, and then also the blood started coming from that particular injury. When the leech is sucking blood from our body, the blood will not be coagulated. Okay, so there is a reason. And also when the leech will fall after sucking the blood, then also from that injury the blood will start coming. Very difficult to stop the blood. Okay, so why the thing is that? Because in the saliva of leech, there are, in the saliva of leech, there is hirudin, which is an anticoagulant. It prevents the blood from coagulation. Clear? Okay. So in the saliva of the leech, there is one anticoagulant, hirudin is there. So which is an anticoagulant? Just I discussed what is anticoagulant. Okay, so it prevents the blood from coagulation. So that is why when the leech bites, the blood cannot be coagulated easily. Clear? So now coming to the next topic. Okay, students, now before we move to the process of blood coagulation, let us have a basic idea on the topic hemostatic plug. What is hemostatic plug? Okay, hemostatic plug. So first understand what is hemostatic plug. Suppose this is a blood vessels. This is a blood vessels. So if suppose this is the inner lining of the blood vessels, clear? So if any rupture is there, then what will happen? Collagen will come out from this rupture, and this collagen what will do? It will attract the platelets. So the collagen will attract platelets towards it. So what will happen? Suppose there are blood vessels and this rupture is there. So the collagen will attract the blood vessels, okay, the platelets. So the platelets molecule will start depositing here, and they will secrete serotonin plus ADP. They will secrete the serotonin plus ADP. Okay. So, and what will happen? And heparin will not work on this. That means it cannot stop of segregation of these particles. Okay, heparin will not work on this, clear? So what will happen? So this is known as the hemostatic plug inside the blood vessels. Okay, so I am describing what happened hemostatic plug. The blood is flowing inside the blood vessels, clear? If due to any injury, what will happen? The collagen will come and it will attract the platelets, and the platelets molecules will start depositing on it, and it will secrete serotonin and ADP. So there is a deposition of that, okay, or sedimentation of that, okay, inside the blood vessels. Okay, so this is known as the hematostatic plug, okay, or hemostatic plug, clear? So what is the hemostatic plug? Okay, so hope you understand it. Okay, so let us move to the next topic. Okay, students, now we will discuss the process of blood coagulation. The process of blood coagulation. Process of blood coagulation. The process of blood coagulation, clear? So the first point, when there is an injury, when there is an injury, when there is an injury, then the platelets will break and it will secrete secrete thromboplastin. Clear? So first what happened? When there is an injury, then the platelets will break and it will secrete thromboplastin. Clear? Number 2, then thromboplastin with the calcium ions in blood, it will create the enzyme prothrombinogen. Prothrombinase. Okay, so it will create prothrombinase. Now, so thromboplastin Ca plus prothrombinase. Now, number 3, this prothrombinase will react with prothrombin and make thrombin. Okay, that means what you can write? Prothrombinase what it will do? Prothrombin thrombin. Clear? Now, what happened? So, first what happened? If there is an injury, the platelets will break and what it will secrete? Thromboplastin. Clear? So, the thromboplastin with the Ca plus ions that is present in the blood, it will create prothrombinase, the enzyme. So, the thromboplastin, Ca plus ion, so prothrombinase. Clear? So, prothrombinase will react with prothrombin and make thrombin. Okay. Now, there is one plasma protein that is known as fibrinogen. Okay, plasma protein is there which is known as fibrinogen. So, there is one plasma protein known as fibrinogen. So, thrombin already created over here. Then next point, thrombin will react with fibrinogen and makes fibrin. Okay, so that means what? Thrombin fibrinogen fibrin. Clear? So, now this fibrin is nothing but a net-like structure. So, with the WBC and RBC, with all the cells, they cannot come out from it. Clear? So, that is nothing but the blood coagulation. Okay. So, first of all, it will secrete the thromboplastin. So, thromboplastin will prothrombin, it will create what? Thromboplastin, Ca plus, so prothrombinase. So, prothrombinase will react with prothrombin and it will create thrombin, and thrombin along with the plasma protein that is in the plasma fibrinogen, it will react, the thrombin will react with the fibrinogen and it will create fibrin. So, fibrin is a net-like structure which will create a guard, okay, of that injury place, and so that the other blood cells cannot come out from it. So, that is nothing but the blood coagulation, clear. So, this is the process of coagulation of blood. So, students, hope you have understand this. So, next move to the next topic. So, students, let us now move to the next topic. Two terms we have heard that. Two terms are there, we have heard that, that is known as CT and BT. So, what is the full form of CT? Clotting time. Clotting time. What is clotting time? So, after injury, it is the time from start of bleeding to the blood coagulation. Okay. So, that is known as the clotting time. What is the clotting time? It is the time from when the bleeding starts, from there up to the bleeding there is a coagulation. Okay, that means the bleeding will stop. Clear? So, that is known as the clotting time. So, it is what? 3 to 7 minutes. And BT means bleeding time. Bleeding time. Okay. BT means bleeding time. So, it is the time duration of bleeding after injury. So, when there is an injury, before coagulation, the bleed will come out, clear, and it will stop, and then the blood will be coagulated. So, this time is 2 to 5 minutes. The bleeding time is 2 to 5 minutes, clear. So, hope you have understood what is clotting time and what is bleeding time. Clotting time means when the bleeding starts, from that time up to the time the blood will be coagulated completely, clear. So, basically for normal injury or small injury, it is 3 to 7 minutes or 3 to 6 minutes in some books. BT means bleeding time. Bleeding time means the bleeding will start and until it stops, clear. Then the blood will be coagulated, clear. So, it will be what? 2 to 5 minutes, clear. So, this is CT and BT. So, hope you have understand this. Okay, students. Now, let us move to the next very simple and interesting point that also many times comes in the examination. That means role of vitamin K in blood coagulation. I already discussed the role of calcium ion in blood coagulation. So, prothrombin, if you can recall the process of blood coagulation. So, prothrombin, okay, thromboplastin, it will react to make thrombin. So, prothrombin is a very important component for blood coagulation, clear. So, now this prothrombin actually generated in our liver with the help of vitamin K. Okay. So, this is the one major component which helps blood coagulation, clear. So, when the platelets will break, then prothrombin will act with that, clear. So, prothrombin will be secreted in our liver, will be generated in our liver with the help of the vitamin K. So, if there is vitamin deficiency of vitamin K, then prothrombin will not be generated. So, what will happen? So, thromboplastin. So, thromboplastin will cannot react with prothrombin because it is not there. Then thrombin will not be created. If thrombin will not created, then fibrinogen. Who will react with fibrinogen? Thrombin. So, thrombin will react with fibrinogen and make fibrin. So, the fibrin then make a layer like net, then that is known as a blood coagulation. So, that process will be, so this is very very important, okay, prothrombin, so that will not be created if there is a deficiency of vitamin K, and that is secreted from our liver, clear, or generated from our liver. So, that is why there the vitamin K has a very important role in blood coagulation, clear. Now, students, another point it is, what is serum? What is serum? Okay. So, serum, suppose the blood coagulated over here. So, after blood coagulation, the yellowish color fluid will come out from that particular place, clear, of the coagulation place of blood. So, that is known as serum. So, the yellowish color fluid which will come out after blood coagulation is known as serum. So, in serum no fibrinogen is there, okay. So, what is serum? Serum means when the blood will be coagulated, then from that coagulated portion the yellowish color fluid like things will come out. So, that is nothing but the serum. So, the serum do not have any fibrinogen in it, clear. The blood plasma protein is fibrinogen. So, the serum do not have any fibrinogen inside it, clear. So, hope you have understand what is serum. So, students, next topic is why citrate salt is preferable to preserve blood in blood bank. So, what we know, citrate and oxalate salts are anticoagulant. That means, if we add this, then the blood will not be coagulated, but it is preferred the citrate salt than oxalate salt. Why? Because it do not have any toxic effect, okay. The citric salt, this salt do not have any toxic effect, and our body cell can utilize this salt into for its metabolism process. So what? No harmful reactions much will be there, because it don't have any toxic effect, as well as our cell, our body cell can utilize the citrate, okay, for its metabolism process. So this is the reason why the citrate salt is used to preserve the blood, okay, in a blood bank. Otherwise what will happen? If we preserve the blood, if we keep the blood, then after some time it will be coagulated. So the anticoagulant substances should be added to it to preserve it for some days. Clear? So which type of salt is preferred? Citrate salt. Clear? So citrate salts are preferred to use than oxalate salt. Clear to all of you? Okay. Now let us move to the next topic. So next keywords, I can say it. Okay. So these are the some keywords you should remember in your NEET exam as well as board exam. Okay. So what is living test tube? What is living test tube? So if we take jugular vein of a horse and take out from its body and tight the two ends of it with thread, then the blood inside it will not be coagulated. This jugular vein with liquid blood inside it is known as living test tube. Clear? So the jugular is a special type of veins. Clear? So if that vein is cut out and it is tight with the thread to end and the blood is there in liquid form. Clear? Then this vein is known as living test tube. Clear? So what is living test tube? If we take the jugular vein cut out from the horse body and if the two end is tight and the blood is in liquid form inside the vein, so that is with the blood inside it is known as living test tube. So I think you can now understand what is living test tube. So let's move to the next topic. Okay students, so now next topic, very important one. This is the last topic for today's episode. Blood grouping system. Okay. So the German scientist Landsteiner in the year of 1900, okay, he basically discovered the different blood groups. Okay, why? So when there is a huge bleeding will be there, then the transfusion of blood or transmission of blood is required. So, the blood, if we see in general, then we see that what happened, that it is red in color, but it was found that when the blood is giving from one person to other person, then sometimes what will happen, the RBC will be what, will be in the solid form, and what will happen, it will just become into solid together and clot will be there and the persons will die. Clear? So, this scientist Landsteiner, on the basis of the agglutination, okay, what is agglutination? It is present in agglutination. It is present in the membrane of RBC, and another agglutinin, it is present in plasma. So, agglutination two types, A and B, and similar type of things, agglutinin, it is two types, alpha and beta. Clear? So, agglutination are of two type, A and B. It is present in the membrane of RBC, and agglutinin, that is present in our blood plasma. Clear? And two type of agglutinin are there, alpha and beta. So, on the presence of this agglutination and agglutinin, okay, the blood are classified into four groups, A, B, AB and O. These are the four groups of the blood on presence of these two particles. Clear? Agglutinogen and agglutinin. Okay. So, on the presence of these two particles, okay, where agglutinin A present or agglutinin B present or agglutinin A alpha or beta is present or they are absent. So, in basis of that, the blood is classified into four groups, A, B, AB and O. So, this process of grouping the blood is known as also ABO process. This process of grouping the blood is known as ABO process. Clear? So, that is the group of blood grouping system. So, in the basis of the agglutinogen which is present in the membrane of RBC and the agglutinin which is present in the plasma. So, agglutinogen two types, A and B, or agglutinin two types, alpha and beta. Clear? So, this is known as the grouping of blood A, B, AB and O, and this is known as also ABO process. Okay. So, this is what the blood grouping system. So, now let us share you very basic information and then I will close this episode today. Friends, now it is our blood agglutinogen, or agglutination and agglutinin. Okay, agglutinogen are of two types. It is what, A and B, and it is alpha and beta. Clear? Now, first number one, in case of A, agglutinogen A is present. Clear? And anti-A or beta agglutinin is present in plasma. It is present in RBC and it is present in plasma. Okay. Now, number two, for B, so agglutinogen will be B. So, it will be what? Anti-B or alpha. Clear? Now for AB, agglutinogen AB will be present. So no agglutination will be, agglutinin will be present. Clear? And from O, no agglutinogen will be present. And here alpha beta both are present. Clear? So see, A can take blood from A as well as from O, because here no agglutinogen is present. So A cannot be given at B, okay, where? Because they will react. Clear? So here no agglutinogen is present, that's why the O can be given to A, and A itself can give to A or donate A. Clear? So same type of agglutinogen or no agglutinogen will be present. Clear? So it will not react. So the B can take from B, because B agglutinogen will be present, or from O, that will be no agglutinogen is present, then that can be given. No reaction will take place. Otherwise the RBC will what? Will coagulate it. Okay. Now AB can take only from AB or from O. Clear? Otherwise it cannot take from any others. Clear? Now O can only take from O, because it don't have any agglutinogen. Clear? So that means what? O can give blood to all. That's why it is known as universal donor. And AB can receive blood from all. That is why it is known as universal acceptor. Okay. So A agglutinogen tinogen A will be present, and beta agglutinin will be present. B will be present agglutinogen, and alpha will be present. AB agglutinogen will be present, and no agglutinin will be present. O no agglutinogen will be present, alpha beta agglutinin will be present. That's why, as it don't have any agglutinogen, that's why it can be given to this, it can be given to this, it can be given to this. Clear? So no reaction will take place. Clear? Because it don't have any agglutinogen. Now AB, both the agglutinogen are present. That's why it cannot be given to all, but it can be take, because if it take from A, then also no problem, because A is there, they are present. If B, so B is also present. If O can be given, then no agglutinogen will be present, no reaction will take place. That's why the AB can take the blood from all other groups. Okay. But O can give blood to all other groups. That's why O is known as universal donor and AB is known as universal acceptor. Clear? So students, hope I can share some very important and very common term, okay, which are very useful for the examination as well as for the entrance exam. The terms, what is living test tube, okay, how the blood coagulation takes place, okay, what is the time of coagulation of blood, what is CTBT, that all in details I discussed in this episode. And what is universal donor, universal acceptor, why, and basis on agglutinogen and agglutinin. Okay, the classification of blood in 4 groups A, B, AB and O, why O can distribute blood to all other groups and AB can receive blood from all other groups, what is the reason. Okay, so that all I have discussed. So if you have any doubt, any clarity you want, you write in comments. I will try to explain that once again for you only. Okay, so see you in my next video. So it will be also uploaded very soon. If you still not subscribe my channel, subscribe it, so to get more and more information on this on different type of topics. Okay, keep watching my channel. Thank you.